Hello, my name is Max Raymond Michalak. It is the 5th of March, 2022. And for my presentation, I'm going to make myself smaller here. I chose to do my presentation on the anatomy and physiology of the retina. So moving on to the next slide. So just first, I want to go over the gross anatomy of the eye. So where is light going to come from? It's going to enter through the cornea, and then we have the pupil, which is going to modulate the amount of light that reaches the retina, and then the lens is going to focus the light onto the retina. And we see the retinal layer right here, and we see the optic nerve. The optic nerve is composed of the axons of the ganglion cells, and then we also see the macula, which houses a pit known as the fovea. And this area is the area of the highest visual acuity. It's the center of the visual fields, and this is housed within the retina, of course. Moving on, so there's a whole lot to unpack here. This is just overall the anatomy of the retina. And first I want to note, where is light entering from? So light is entering from the vitreous layer in between that of the lens and the retina. So we see the light entering, and you would think maybe the process of the electrical, or the light energy being converted to electrical energy, you'd think it might start where light hits first, on this side, on the anterior side of the retina. But really, it doesn't start there. It starts, phototransduction is what that process is denoted, starts at the photoreceptor cells, the rods and the cones. But before I talk about that process and what cells go to what cells, I first want to note the peripheral side of the retina. We have the pigment epithelium, and this epithelium sits behind the photoreceptor cells, and it serves to absorb any light that the photoreceptor cells do not absorb. So it absorbs any of that light, and it prevents that light from reflecting off and bouncing throughout the eye and causing distraction. So it absorbs that light. From there, we can see these photoreceptor cells. We can see the rods. We can see the cones. And here we have a bunch of rods here. But the rods and the cones, then they pass on information to the bipolar cells, which pass the information on to the ganglion cells, which then, their axons, as I said, make up the optic nerve, and the sensory information is provided to the brain where it can be processed and allow for sight. There are also, we see two other different kinds of cells. We see horizontal cells and amacrine cells. So horizontal cells actually modulate the synapses between the photoreceptors and the bipolar cells, while the amacrine cells modulate the synapses between the bipolar cells and the ganglion cells. And then, since this is neural tissue, we also have those support cells like glial cells and astrocytes present, although they're not seen within this diagram. But over to our right, I just want to touch on it for a second because we're going to go over it a little bit more in detail in the next slide. But convergence is a concept or a process that occurs within the retina, and it is the process where you have multiple neurons converging onto a single neuron. So in this case, we have 3 rods converging onto one bipolar cell, and we have 2 bipolar cells converging onto one ganglion cell. So overall, we had 6 photoreceptor cells converging onto one ganglion cell through the process of 2 bipolar cells. So moving on to the next slide, so convergence of the retina. So the areas of lowest convergence, so a 1:1:1 ratio. So 1 here, 1 cone, 1 bipolar cell, 1 glial cell, or ganglion cell, my apologies. This is found in the fovea and the macula. As we said, the area of highest visual acuity is going to have the lowest convergence. Well, on the other hand, as we move further away from there, towards the periphery and the edge of the retina, we are going to have more convergence observed, with more photoreceptors converging onto one bipolar cell, with multiple bipolar cells converging onto one ganglion cell. And this is going to be the area of lower visual acuity, where it's not the center of the visual fields as we see in the fovea and the macula, but it's just going to be lower visual acuity. Moving on, we have photoreceptors and visual pigments. So now kind of looking specifically at the photoreceptors themselves. So here we see the rods and the cones. As I said, these are our two photoreceptors, our two primary photoreceptors. So they all have an outer segment, an inner segment, and a basal segment, or a synaptic terminal, as it's labeled here. So the outer segment houses these discs, which also, within these discs is housed the photosensitive molecules, or the visual pigments. And then in the inner segments, we have the organelles, we have the nucleus, and then the synaptic terminal, we have where glutamate is released onto the bipolar cells. So rods are used for black and white vision. They usually outnumber cones, according to Silverthorne, I believe 20-1. But they are found in the fovea, where cones are only found in the fovea, and they're for high acuity vision and color vision. So as far as pigments, rods have the pigment rhodopsin, where cones have 3 separate pigments, but they all function very similar to rhodopsin. So in the coming slides, we're going to use rhodopsin as our example. But there's 3 different kinds of cones in that same respect: red sensing cones, green sensing cones, and blue sensing cones. And if you have a defect in one of these, you could actually have color blindness. I actually have red-green color blindness, so I likely have a defect in my red and green sensing cones. So moving on. So signal processing, the process of transduction. We're going to start with rhodopsin. As I said, it's going to be our example here. And rhodopsin is made from opsin, a protein embedded within the disc membrane of the rods, and then also retinol, which is a vitamin A derivative, and it's the light-absorbing, the photosensitive molecule that when light hits it, it is going to dissociate from opsin, and it's going to eventually enter into the pigment epithelium. But this process is called bleaching, where the conformational change is seen in rhodopsin. So how does this bleaching allow for phototransduction? So this process is based on 3 different cation channels. We have CNG, which is cyclic nucleotide gated ion channels. We have potassium leak channels, and then we have voltage gated calcium channels. So CNG channels, or cyclic nucleotide gated channels, are going to be allowing for the influx of sodium and calcium into the rod, while potassium leak channels are obviously going to be allowing for the slow leak of potassium out of the cell. And then voltage gated calcium channels are going to be found in the synaptic terminal, and they're going to help modulate the amount of glutamate that is released onto the bipolar cells. So in conditions of no light, where it's completely dark, cyclic GMP is high within the rod, and therefore the cyclic nucleotide gated ion channels and the potassium leak channels are both open. So we're having an influx of sodium and calcium into the cell, and we're having the efflux of potassium out. This allows for the resting membrane potential of the rod to be at about -40 millivolts. And at this voltage, the voltage-gated calcium channels are all open, and we're just allowing for the release of glutamate into the synaptic cleft onto the bipolar cells. However, if light is present and rhodopsin is activated, meaning that retinol dissociates from opsin, has that conformational change and dissociates from opsin, light, this bleaching process, that change in rhodopsin, or the release of retinol, causes a secondary messenger cascade to initiate. This is through the protein transducin, which is G protein, and this decreases the cyclic levels of GMP within the rod. This decrease in cyclic GMP then closes the cyclic nucleotide gated ion channels. So calcium and sodium are coming into the rod at a slower rate, while the potassium leak channels are still open. So this causes a hyperpolarization. And therefore, this hyperpolarization from about -40 to -70 causes a decrease in the overall glutamate released. And that's in air. This is in extraordinarily bright light. All rhodopsin would be activated, and all the CNG channels would be closed, and this would inhibit all glutamate release. You'd have no glutamate release at all. But in dimmer lighting settings, we're going to see this graded effect, where depending on the amount of light exposure, will affect how much glutamate is being released, with bright light having less glutamate released, and dimmer light having more glutamate released onto the bipolar cells. So following activation of rhodopsin, we then move on to the bipolar cells because we see this process of how glutamate's released from the photoreceptors. So now there are two different kinds of bipolar cells. There's light on and light off. In this diagram, they're respectively called on-center and off-center bipolar cells. But light-on bipolar cells are inhibited by glutamate release in the dark. So in the dark, where there's being a bunch of glutamate released from the photoreceptor, this is going to inhibit and decrease the amount of glutamate that's released from the light-on bipolar cells. But the light-off bipolar cells are actually excited by this amount of glutamate. They like this dark environment, or I shouldn't say they like, but they will release more glutamate in these darker conditions when they are excited by more glutamate from the photoreceptor. And this whole process goes in the opposite direction as well, where light-on bipolar cells are excited by less glutamate in the light. So when the light's present and the photoreceptors are releasing less glutamate, they're actually excited and will end up releasing more glutamate. But light-off photoreceptors in light are receiving less glutamate from the photoreceptor, and then will subsequently be inhibited, and they will release less glutamate. So we see this graded effect from the photoreceptor to the bipolar cell to the ganglion cell of how much glutamate is being received initiates a response, a respective response by the following cell. So from here, we see that we go from the photoreceptor cell, then we go to the ganglion cell, and we see this release on the ganglion cells, and then this information can then be carried to the brain and processed there. And then here's my sources, and that is all. Thank you.